Martha Chase helped chase down the source of genetic information in the Hershey-Chase or Warring blender experiments she performed with Alfred Hershey in the 1950s. And these experiments helped show that nucleic acids like DNA and RNA are the source of genetic information and not proteins. A lot of people thought it would be proteins because proteins have more like complexity. They have like 20 letters, DNA only has 4. The letters of proteins, amino acids, they have all these different properties, whereas DNA letters look pretty similar. But in their experiments, they were able to cleverly show that it was the DNA that was passing on the genetic information. And they did this using viruses that infect bacteria, so bacteriophages. Basically, scientists knew that these phages would attach onto bacteria, inject something into that bacteria, and then the bacteria start making more phage. So clearly the phages were injecting some sort of genetic information that was telling the bacteria how to make more phage. But what was this makeup of that information? That was what was not known. So what Martha and Alfred did was basically they did some clever labeling. So they were able to label the nucleic acids of the phage and the proteins of the phage, and then they were able to track down what was injected into the bacteria. So basically they labeled the nucleic acids, or they labeled the proteins, and then they got the phage to inject this stuff into the bacteria. And then this is where the blender comes in. They use the blender to shear off any of the phage that had injected anything, so any of the stuff that's left on the outside, and then they look to see where the radioactivity was. When they labeled the proteins, they found that the radioactivity stayed in the stuff that was blended off, so that in the capsid and the shell of the protein. They call it the ghost. Whereas when they labeled the nucleic acids, now that nucleic acid was going into the bacteria, and so it stayed in the stuff that wasn't getting sheared off. So the experiments weren't exactly that cut and dry. It was only 80%. But so it didn't quell all the critics, but this was building upon a lot of earlier research as well. And I think that this one kind of is remembered more because it's got that blender coolness. But speaking of being remembered, Martha Chase is not that well remembered. Alfred Hershey didn't even mention her in his Nobel Prize acceptance speech, even though that this was the work that she and he did. But she was just his research assistant, although she is cited on the paper and stuff, and so people think she did a lot. There was a lot of sexism going on. She has this pretty tragic history, but there's really not that much known about her because she was kind of forgotten by history largely. So I want to tell you a little more about her as well as the details of this experiment now that I've gone over the basics, because I like to give you the details and we'll look at some of the actual figures and that sort of thing. So let's dive in.
So this is where we're going, but let's set the scene first. So at this time, people were trying to figure out what it is the genetic information that, what type of chemical molecule is storing genetic information. So we know that genetic information exists, but how is it really getting passed on? What's it stored in? And so this is coming after in a context where there were some very key experiments before this that they built upon. So it was found by Griffith, and Griffith's is hard to say. Griffith's experiments showed that you could take bacteria, and so there would be this S strain and this R strain, and the live S strain would kill mice. But if you killed the S strain, then the mice lived. But if you took that heat cell killed S and mixed it with live R, and so R is normally not that doesn't kill the mice. But if you add this S, it can transform, it can somehow pass some sort of genetic information to this harmless strain or less harmful strain. And then this is going to somehow transform this R into S and make this a dead mouse. And so something in the S cells can transform the R cells into S cells. And so this was found by Griffith, that there was some sort of transforming substance, and then Avery, MacLeod, and McCartney decided to find out. And they really deserve a lot more credit than they've gotten too. But I think part of this was that we're coming at a time when people just really didn't believe that DNA could be holding this genetic information because protein seemed way, way, way more likely. But I have a whole post on their paper and stuff, but was happening in 1943, and basically they added things. They wanted to see what it was to that in the S that was transforming the R into S. And so basically they mixed things, but they added additional things to remove various components. So they added proteases, so like to chew up the proteins. They added lipid chewers, they added polysaccharide chewers, they added RNA chewers and DNA chewers. And they found that only when they added the DNA chewers did the transforming principle go away. And so this really helped show that DNA was this transforming substance, but people still didn't think it was conclusive enough. And at this time, especially, there was a lot of resistance. And so this is setting the scene. And now let's talk about how Martha and Alfred Hershey went about figuring out that and showing that DNA really was the thing, although even their results weren't entirely conclusive. And there's some kind of, there's a bunch of like history people trying to figure out like, well, why did they get accepted more than others or whatever. I think part of the reason why we remember it so much is because they used a blender, and blenders are cool. And now I hope that you'll also remember it because it's Martha Chase. Okay, so let's talk about this Hershey-Chase experiment. It's actually Hershey-Chase experiments. So they have this paper, and I'll take you through some of the key figures and findings. This was published in 1952 in the Journal of General Physiology. And it's got this kind of unassuming name, and it's dependent functions of viral protein and nucleic acid and growth of bacteriophage. But it's actually really interesting. And in addition to doing this, this classic blender experiment, they actually do a lot more experiments, and I'll take you through some of those as well. But they're going to go about this using this phage system. So a phage or a bacteriophage is a virus that infects bacteria. And there's this T2 phage, and basically it docks onto bacterium and then it injects stuff. Then the bacteria makes more phage, and the bacterium bursts open, so we call it lysis, releasing this new phage. And then the phage injects more bacteria, and this cycle continues and continues. So they basically, they do experiments that are going to show that this protein, confirm that there's a protein coat surrounding DNA, that the DNA gets injected, and that the protein stays out, and that it's the DNA, therefore, that is providing the information for making more phages. And so I want to tell you about how they're going to do that. In order to do that, they need to be able to track the protein components and track the DNA components. So DNA and proteins are both made up of atoms, like everything else that matters. And so basically, atoms are like the smallest units of a particular element. So hydrogen, carbon, nitrogen, oxygen, and they have this makeup with the subatomic particles where they have this nucleus where they have a high concentration. There's like protons, which are positively charged, and then neutrons, which are neutral. And then there's this cloud of electrons around them. And hook them up in different ways, and you get different types of molecules. So you hook nucleotides up, and you get DNA and RNA, which we now know have the instructions that are going to tell the ribosome making the protein making complex to link together amino acids, which are protein letters, to make proteins. But the key thing is that all of these are made up of atoms, but not the same types of atoms. So DNA and proteins, they have a lot of the same atoms. They have a lot of carbons and hydrogens because, well, that's the backbone of organic chemistry, this like carbon-hydrogen backbone. They also both have oxygen and hydrogen, but proteins also have phosphorus, and you're not going to find phosphorus in amino acids. And so unless you're talking about like you have a kinase at a phosphate group, but we're not talking about post-translational modifications here. But 2 amino acids, so there are 20 common amino acids, and 2 of them have sulfur, and no DNA has sulfur. So this phosphorus and the sulfur, These are the things that are unique to DNA and unique to proteins. And so if we can label these, then we can selectively track the DNA versus the proteins. And we can track them using radioisotopes labeling. So basically, we talked about the atoms have those different components, and the protons is the number of protons is what defines an element. So hydrogen has 1, nitrogen has 7, carbon has 6, oxygen has 8. And so they're always going to have that number of protons, but they can actually have different numbers of neutrons and different numbers of electrons. If you change the number of electrons, you're going to get a charge because the electrons and the protons, the number needs to be balanced in order for a molecule to be neutral, since the electrons are negative and the protons are positive. But if you change the number of neutrons, since they're neutral, you're not going to change the charge. But what you might do is you might change the stability of the nucleus. And so there are some, if you add or take away neutrons, you might make the nucleus more unstable. And when it's unstable, it can get unhappy and it can undergo radioactive decay in ways that are going to let off radioactive energy. And this can then be detected. So there are different radioisotopes for different elements. And the one I use a lot in the lab is this P32, which is the radioactive phosphorus. And it undergoes this beta minus decay. And then they're going to use this radioactive sulfur S35. So we have P32 and S35. So going back to our labeling, we can use P32 to have to label the DNA, and we can use the S35 to label the proteins. So if we go back to that cycle of life for this phage, basically they know that the bacteria are going to make more phage if they let a phage infect bacteria. So what they can do is that they can make the bacteria make labeled phage if they grow the bacteria in media, so in bacteria food that's containing radioactive phosphorus or radioactive sulfur. Now, when the phage injects into the bacterium is going to make more phage, but now it's going to be labeled phage. And then the bacterium is going to burst, it's going to lyse open and release these phage particles. And then they can isolate out these phage particles and then use them to infect other bacteria, so they can kind of have better control over this process. They can do this in media that has the labeled phosphorus or media that has the labeled sulfur, and then they can get labeled phage made. And so this is what they're going to do and use as this key tool in their experiments. So we'll get to those blender experiments, but as I mentioned, they did a lot of other experiments, and the blender experiment is actually like towards the end. So they need to build up a little bit of stuff first, and it's kind of cool how they do it. I'll note that the paper is fairly complicated and technical, but they explain things very well. But I'm not a microbiologist, and so I hope I get everything right. But here's my stab at it. So basically, one of the first things they did was to confirm this other scientist, Thomas Anderson's findings, that a phage seemed to be able to break open and separate the protein shell in free DNA. So we're talking here about the phage particles lysing, not the bacteria lysing. So when we talk about lyse, just means to break open. So the phage is going to make this bacteria break open the bacteria lyse, but in this case, the phage is staying intact. But it was found by Anderson that these phages themselves could actually break open and separate into a protein shell, which he called a ghost, and free DNA. And so basically in this experiment, what they did was they took that labeled phage. So they labeled phage with the P32, which is for the DNA, and the S35, which is for the protein. And they either kept it as is, or they lysed it by osmotic shock. And so basically osmosis is when you have a semi-permeable partition between two things, and you have like water moving between them, and so, or whatever your solvent is. So water can get through, but other things can't. And so basically the water is going to diffuse through the membrane into the region where there's more solute. So more on this in other posts. But you can think of it as kind of, it's not really like this, but it has the effect of kind of like diluting out the solute. So basically there's a lot of stuff. Solute is just something that's dissolved. And so there's a lot of stuff inside of the phage particle. If you put it in a lot of like dilute water, like just water, without a bunch of stuff, water is going to flow into the phage, and this can make it like get like, because you have too much water going in. So basically what they did was that they suspended the particles in high concentration of salt, so make it really salty, and then rapidly dilute the suspension with water. And so this is making this like osmotic shock. So they could do this with their DNA labels or the protein labels. And now they want to basically see where the DNA is and where the protein is. And so in this case, we're lysing the phage. We're no bacteria here, just bacteria involved in the actual making of the phage, but we're not considering the bacteria in this case. So what they're looking at is this acid soluble fraction. So to the best of my knowledge, this is like small pieces of DNA, or like individual letters. So if you take DNA and just like naked DNA and you were to treat it with DNase, which is a DNA chewer, then you're going to get acid soluble DNA. But if that DNA is protected from the DNA chewer, then you're not going to get acid soluble DNA. So they took the whole phage, so that wasn't lysed, and the lysed. And what they did was they initially just looked at the acid soluble as is, and they didn't see, so I mean, one, I don't even really know what one course means, but if it means anything, but they couldn't even measure it in most of these cases. But when they added DNase, now they found they still really didn't see much of anything in the whole phage. So the DNA was protected in this phage, but it was vulnerable to degradation after lysis. So basically, they're showing that after this is lysed, this DNA is removed from its protective part. Now they want to look at absorbance to bacteria. And so basically they found that the protein shells could stick to the bacteria, even if you chewed up the DNA. So here, when you have a whole phage, you have both the radioactivity is going to be stuck to the bacteria, even if it's in, no matter what it's in, because the phage, if the phage is stuck, then you're going to see radioactivity because the stuff that makes up the phage is labeled. But if you break that phage, and now you have free DNA and you have this protein coat, now the protein coat is going to stick to the bacteria, but the free DNA is not. So, and then finally, they found that these anti-phage antibodies could detect the protein, but not the DNA. So when they have this anti-phage, they can use it to kind of like precipitate out the phage, and then they see where the radioactivity is, and they can see that the radioactivity, as detected by these phage antibodies, is detecting the protein. And so what this is saying is that the protein is on the outside, so it's accessible to these antibodies. And so their key conclusion is that the ghosts represent protein coats that surround the DNA of the intact particles, react with antiserum, protect from the DNA from DNase, and carry the organ of attachment to bacteria. So the protein's doing the job of protecting the DNA. It's on the outside. It's doing the job of docking onto the bacteria as well. So we know that the protein can stick to the bacteria, and that if you lyse these phage particles, you can get the DNA to separate out. But what about when you're just like infecting bacteria? Now what's happening to the DNA? They also did some kind of complicated experiments. So I have to say that this part I'm like the least sure about. But they were doing basically experiments where they're showing that if they can sensitize the DNA, so they can get it to come out of the phage. And so before we had seen about how they could do that just by lysing the phage. But they also show that when this is actually happening in bacteria, the DNA is getting injected or it's getting protected from the DNA's chewer as well. And so it's when they get the cells to break open, it seems like you still have significant protection. So it seems like there's some sort of like shells inside of the DNA when the phage is getting made, but that the phage DNA is going to be protected inside of the cell. And if you break open the cell, then you can get some of that, but most of the phage DNA is going to remain inside of the cell membranes, as do the mature phage progeny. So it seems like we have the shell and then we have the DNA, and we know that the DNA is getting into the cell. So we build up these pieces, and now we're going into those classic blender experiments. And so here they're going to label the protein, or they're going to label the nucleic acids like we saw before. But they're going to do so in a way that what they're going to do is they're going to actually see what is getting into the cells and what is staying outside of the cells, and therefore, and what is like getting passed on type of things. So they could selectively label the protein or the nucleic acids like we saw before by growing them in the bacteria in the labeled medium, and then using that phage the labeled phage that they created to inject more bacteria. And they could do this with nucleic acid, the phage labeled with nucleic acids, or the phage labeled with proteins. The key thing here is that what they're going to do is the reason they use the blender is that it's going to be used to shear off the phage cell. So they showed that there was that like phage shell, the protein phage shell that seems to be attaching to the bacterium. And now we want to see, okay, well, if we remove that shell, what remains in the cell? And so they're going to use the blender to kind of shear off that stuff without breaking open and disrupting anything else. So centrifuges go really, really fast. So they'll centrifuge it later, but they don't want to risk like doing other things with the centrifugation. Instead, they just want to do something gentler and kind of strip the cells from the outside stuff. So you can imagine, if they label the protein and the radioactivity was, if they label the protein and the thing that was actually like going in was protein, then they're going to see that the radioactivity is in the bacteria. If they label the protein, but the stuff that's actually going in is the nucleic acids, now that radioactivity is going to be in that shell that they're shearing off, and there's going to be no radioactivity in the bacteria. If they label the nucleic acids, now if the genetic information is actually coming from the protein, if it's the protein that's going inside, then they're going to see no radioactivity in the bacteria because the protein isn't labeled, but they'll see the radioactivity in that shell that they shear off because they're going to collect the different fractions, don't worry. So they collect this part, they take off in the part that stays in. And if they label the nucleic acids and it really is the nucleic acids going in, now your radioactivity is going to be in the bacteria. So this is what they did. So they do nucleic acid labeling. And when they do this, they see that, so they take this radio-labeled phage, this has its DNA labeled, and then they see that they do this shearing to remove the phage cell, then they centrifuge it to separate the phage shells and the bacteria. And what they measure the radioactivity. And the phage was not radioactive, but the bacteria was radioactive. And when they do this with protein, this case, because the protein is labeled and you're shearing that off, now your phage is going to be radioactive, but the bacteria is not radioactive. So by phage, I just mean like this phage shell. So the results weren't as cut and dry as that might seem. Nothing in science is ever quite that clean, but they found that the protein was staying out mostly, or at least mostly in terms of how they measured it. So they found that about 80% of the sulfur was staying extracellular, whereas the DNA here only about 25-ish% was staying extracellular. And so most of the DNA was going in, most of the protein was staying out, or at least it looked like most of the protein was staying out. And the important thing too is that they're taking bacteria as well. And so they're taking bacterial samples and measuring the amount of phage. And by doing this, they're actually seeing that this actually, the cells are being infected. And so this is corresponding with the infection process, is this phage part of the phage going in, the DNA part, and the protein staying out. So some of their key conclusions were that basically the 75% to 80% of the phage sulfur can be stripped from these cells using their blender method. This release of the sulfur happens, but only 21 to 35% of the phosphorus. And the treatment does not cause any appreciable inactivation of the intracellular phage. So they're still making active phage. So these facts show that the bulk of the phage sulfur remains at the cell surface during infection and takes no part in the multiplication of intracellular phage. The bulk of the phage DNA, on the other hand, enters the cell after absorption of phage to bacteria. But what about that remaining like 20% of sulfur? And so remember, there's a lot of people that are really rooting for protein to be the genetic information. Could a small amount of protein be getting in and directing the show? So here what they're going to do is they're going to look at the progeny phage. So they infected bacteria with protein-labeled phage. Then they use their blender to remove any of the extracellular stuff. And then they're going to promote lysis of the bacteria and look to see if the new phage particles are radioactive. So basically, there are ways that you can promote the bacteria to lyse, so the bacteria to break open. So remember that bacterial life, in the phage life cycle, the phage is going to get the bacterium to burst open. There are ways that they could do. They can add like UV inactivated phage to basically convince these, promote the lysis, and so get the phage to be released. So if something is acting as genetic information, it needs to be being passed on. So if you have your genetic information labeled, then some of the progeny should have that labeled information. And so what they can do is they can label the DNA or they can label the protein and then look to see if the progeny has radioactivity. And so they show for the sulfur. I couldn't find it for the phosphorus, but they talk about the phosphorus. But basically their technique here was that they're going to infect the bacteria with a protein labeled phage, use their blender to remove any extracellular stuff. So anything that's left on the outside should get removed. And then they can promote lysis of this bacteria. So basically get the bacteria to release all of those phage particles that it has just made. So you get the bacteria to, infect the bacteria with this labeled phage. The bacteria is going to make more phage. You blend off this stuff that's left outside. And now you see if this new phage that was made has the radioactivity. If there was radioactivity that was actually going in, and this radioactivity was important for making the progeny or whatever, you should see some of the progeny should be radio labeled. And you should see that if you remove the coat, if you do the blending part, then the radioactivity should be increased, enriched in that portion, as opposed to in the portion that was stripped. If the genetic information is labeled and it's getting in, then you're removing the stuff that's out, then you should be enriching for this radioactivity. So they can use a centrifuge to separate out these portions, and then they could see where was the active phage, so they can measure the phage, and then where was the radioactivity. And so basically what they found was that the S35 is not, most of it is just going to be eluted in this blender fluid, and that in their centrifugation portions, if they don't strip the cells, the portion with most of the phage is like this high-speed portion. You can see that if they don't strip the cells, about 10% of the sulfur is in that portion. 10% of this radioactive label is in the portion with the phage. But if they do strip the cells, then only about 0.7% is in there. And so this seems to be like an artifact, and not something that's actually needed to pass on this genetic information. And so if they strip these cells, less than 1% of the radioactivity was in the fraction with the majority of the phage. And so this is pointing to the fact that this genetic information, the thing that's actually getting passed on to tell these bacteria how to make phage, and then so these phage can then tell more bacteria how to make more phage, that this information is not coming from this protein portion. So they also said that when they did this with radioactive phosphorus, now 30% or more of the parental phosphorus is going to be contained in the portion that had the progeny phage particles. So the DNA was getting passed on, but this protein part was not. So they did some experiments to try to figure out like what were the properties of that lingering sulfur. And so they did various things where they tested how it stuck to various things. And basically it just seemed to be something that was non-specifically, it wasn't important for the genetic information transfer, is the big takeaway. Another cool experiment that they did, which I don't really show much of, but they did a formaldehyde inactivation to inactivate the phage, and they found that the phage would still stick to the bacteria, but that it's not gonna actually inject. So they know that the inactivated is not able to get the bacteria to make more phage, and they found that it also is not able to inject the DNA into the cells. And so that was pretty cool. And so it seemed like there was some sort of active process that was involved in getting this DNA injected into the cells. So their key findings, they summarize things nicely. So some of the parts of the paper I found kind of confusing, but I really appreciated the summaries. So basically they found that the sulfur-containing protein of the phage particle evidently makes up a membrane that protects the phage DNA from DNase, comprises the sole or principal antigenic material, and is responsible for the attachment of the virus to bacteria. So that's what we're looking at in this experiment. Suspension of the cells agitated in the blender for at least 75% of the phage sulfur and only 15% of the phage phosphorus. And the cells remain capable of yielding phage progeny. So that's what we're seeing here. And looking at the progeny here. The facts data show that most of the phage sulfur remains at the cell surface, and most of the phage DNA enters the cell in infection. Whether sulfur-free material other than DNA enters the cells has not been determined. So the properties of the sulfur-containing residue identify it as essentially unchained membranes of phage particles. All types of evidence show the passage of phage DNA into the cell occurs in non-nutrient medium under conditions in which other steps in viral growth do not occur. They also show that the phage progeny yielded by bacteria infected with phage labeled with radioactive sulfur contain less than 1% of the parental radioactivity. So this is what we were seeing when we were looking at this part, that less than 1% of that progeny phage was actually had radioactivity. The progeny of phage particles labeled with radioactive phosphorus, however, contain 30% more of the parental phosphorus. Okay, and then the phage inactivated by the dilute formaldehyde is capable of absorbing to bacteria, but doesn't release this DNA into cells. And so this is what we're talking about, I was showing you in the paper a minute ago. And then the sulfur-containing protein of the resting phage particles is confined to a protective coat that's responsible for the absorption to bacteria and functions as an instrument for the injection of phage DNA into cell. This protein probably has no function in the growth of intracellular phage. The DNA has some function. Further chemical inferences should not be drawn from the experiments presented. Although we can make inferences, and we know that their inferences, the inferences that were drawn, we now know were very instrumental. So let's talk more about Martha Chase. So there are a couple of things I want you to take away today. So we're going to talk a lot about the science, and that's really cool, and that should be remembered. But I also really want you to remember that that Chase in the Hershey-Chase experiment stands for Martha Chase. So it's really actually, it was really hard to find information about her when I was originally making a post on her. And she has this really just like kind of tragic story. So she was born in 1927 in Ohio, and she got a bachelor's degree from the College of Worcester in 1950. And then she went to work as a research assistant for Alfred Hershey at Cold Spring Harbor Laboratory, CHSL, which is where I am. She resigned in 1953 and then went to work at Oak Ridge National Laboratory in the University of Rochester. And then she would come back during the summers for the phage group meetings. And after almost a decade of working as a research assistant, she decided to return to school. She moved to California and went to the University of Southern California, where she got a PhD in microbiology in 1964. But then she had just a series of tragic setbacks that ended her scientific career, and then she moved back to Ohio after graduating to live with family. And the last few decades of her life, she had dementia, which affected her short-term memory, and she died of pneumonia in 2003. The really tragic thing is how she was treated and how she is remembered, and just that there's not that much information about her. And the information that I can find about her is kind of like very sexist. So Vaclav Zabalski, he just, you can read his transcript of an interview. So he's like described as her friend in like her obituary and stuff, but the way that he was talking about her is just very sexist and gross. I remember Martha Chase as a really great scientist. Fortunately, it's harder to remember her as a scientist when she is not given as much credit as she deserves. So Alfred Hershey actually got a Nobel Prize for doing this work. And he, in 1969, he shared it with Salvador Luria and Max Dilbruck for the discoveries concerning the replication mechanism and genetic structure of viruses. So Martha was not included, and Hershey didn't even acknowledge her contributions in his acceptance speech. And so it's not like, so she was, a lot of times like research technicians and stuff are often like not given their due credit. But the fact that she was, and sometimes research technicians are often not put on to papers because they're often doing work that's not like considered to be like intellectually or substantially contributing, but more just doing the cell culture, doing the work that they are instructed to do by other scientists because that's their job. We often use we use them to do the work that we don't want to do. Thankfully, they get paid hourly, whereas we don't. But basically, research technicians are very important, and sometimes research technicians can take more active roles in research. It all depends on the lab setting and what the arrangement is. But it certainly seems that Martha was taking a more active role because she was included in this authorship of this key paper with Alfred Hershey. And this is especially at this time, that was not common to include your research assistant. And so she really is thought to have played very key role in these experiments and made significant contributions and really deserves credit. And even if she didn't, seriously, Alfred Hershey, you couldn't mention her in your speech. I would like to just give a huge thanks to some awesome lab techs who have made my research possible. So this is John and Soraya, who have since moved on to grad school and other work. and Sumati and Matthew, who are in our lab now and are doing awesome. Yeah, so this is Lovely where Joshua Tour, Culture Hopper Laboratory. But I just wanted to say a huge thank you. So even if they're doing the things that we ask them to do, they're doing above and beyond. They're troubleshooting, they're making things work that we couldn't get to work. They're doing so many great things. And so thank you to them and to all of the other lab technicians out there. I appreciate you and you deserve more credit. Okay, but anyway, so I hope you remember Martha Chase. And so you can learn more about her in this post. And just remember that the Martha in the Hershey-Chase experiments, that Chase is for Martha Chase. And so sometimes when we do this, like we go by the last name, like you don't know, like, oh my God, this is this fascinating woman. And so like Michaelis mentioned, that's Maud Menton. So yeah, so there's some really key women in science that kind of you don't even know did this work. And so yeah, so I will get off of my little like soapboxy thing here, but I hope that you remember Martha Chase. Thank you.